My name is Sivan Sasamoedo. My research is on finding the genetic association between transcription factor 7 like 2 and then type 2 diabetes. We did some comprehensive search on some genetic predisposition to type 2 diabetes because we believe that not only environmental factors that causes type 2 diabetes, we believe that some genetics also play a role in it. So by comprehensive search, we realized that the transcription factor 7 like 2 polymorphisms has been constantly associated with type 2 diabetes. So we did a systematic review and a meta-analysis, and that stood out. So we tried to replicate the association in the Ghanaian population to see the transferability of that SNPs in the Ghanaian population. So the design of the research was such that we took a case control study design where we selected type 2 diabetes patients from the Eastern Regional Hospital, and then we selected controls who are apparently healthy, as we assumed. And then some of the tests we did to exclude are controls not assumed to have anything to do with type 2 diabetes is those that had glycated hemoglobin at a higher or at a rate or at a value that is in the pre-diabetes or in the diabetes state were excluded, and then those that were having also glucose concentration at a value were also excluded. And then we also try as much as possible to exclude those with highly elevated blood pressure. And then after collecting blood samples, we divided it into two, serum separation, and then we stored part of the whole blood. The part of the whole blood was used for actual DNA extraction, and then the genotyping using the T-ARMS genotyping technique. Actually, it's a manual method, and then it amplifies in a complementary manner. And in as much as it's amplifying in a complementary manner, it amplifies both a control, or it amplifies both the SNP and then the allele of choice. So at the end of the day, we're able to, in one reaction, more like a multiplex PCR system, we're able to genotype those who were having the SNP and those who are not having. And we use the logistic regression analysis to evaluate the associations, and then realize that the SNP has a high penetrance in the Ghanaian population, especially type 2 diabetes population. At the end of the day, we were able to replicate the association, which was relatively higher in type 2 diabetes. And we were able to conclude that the SNP is actually associated with type 2 diabetes in the Ghanaian population. So what we recommended was a multi-center research in a high population, and also do inter-ethnic variations in that SNP. And we also recommend functional analysis in the Ghanaian population to see the actual role the SNP plays in predisposing people to type 2 diabetes.